Yes, good morning to all those who are in online as well as in offline mode. Shall we wait for 2
Good morning,
minutes?
sir.
So other can join, right? So on Monday, Aditya sir has taken your lab, right? On a Monday. On this Monday. He has taken some experimental part. Theoretical. Enzymes. Yeah, no, okay. It's theoretical. This part is. Okay. I think basic theoretical part is not started. Oh, no, that is a paper. That is I'm telling. Enzymology paper is started. Okay. Syllabus. That's right. Everyone has their own way to taught. Here it is like that I'm directly giving the some of the parts. Right. That is majority taken from the books itself. That is up to the student mentality. Sometimes if I'm taking in a higher level, everyone is not able to maybe understand. That's also their capacity and level. So mediocre level one has to follow as a teacher. If Just what I am going to take in a master's, I'm going to put before you, you won't be able to digest.
When we go for master law, you take like health sector, those health sector, microbiology.
Medical microbiology.
What all we study this?
What are the subjects will come? Then the clinical pathology, we will be having the at least 2 to 3 paper more. That will be a first priority. Then what are the diseases are there, and what are the diagnostic and the treatments? Each of them is required to, starting with the prevention. What are the preventive methods could be possible? If it is happened, then what are the diagnostic methods are available that you can develop or improve? And then after it is at the treatments. Three are there, and what are the general diseases are there? So one semester, 4 paper, 2 to 3 papers is going to like that only. Other the culturist method. If it is there, how to culture it? This but obvious. Here and there in throughout the world, wherever you go, it will be fall in this category one. That's it. Yeah, then the university and college having their own way to give the one or another kind of papers. Basic papers may be also there, but the core part will be fall in this part. Yeah. Right. So good morning, everyone. I have kept the attendance link for you people, so you can have the same thing to be get open here. You can put your attendance over there. Okay, so the topic that I have started from the earlier also, we will move ahead. That carbon elements till the NaCl, yeah. We have seen up to the salts, hyper and hypotonic solutions up to that we have seen, right? Let's move further ahead. That is like gelatinous, generally obtained from the bones and cartilages from the animals also. Here, the many peoples are having the issues whether it is a veg, non-veg, or the source is a animal tissues, or it is a plant tissue. Sometimes it is mandatory. The reason is what? If you are looking for some virus culture, right? The important phenomena will come in a way that when you are looking for any viruses do require their living area or the capacity, which is as suitable or as such as an host system. And it is always difficult to mimic the nature. In exit area, when you are looking for the food or the nourishment for the microbes, it may be easier. But when you are looking for the certain specific kind of pathogens, or they are the indigenous, sometimes some pathogens are living inside your stomach or the midgut. Within the midgut, such environment to create, it is really difficult. Okay, so in this case, we require to sometime infect or inject such bacterial pathogens or the viruses to any particular host. It is like we are doing certain experimentations on like a guinea pigs sometimes, or in a rats or a rabbit. These are the 3 experimental animals. Other also are there. Then the goat. As you are going into higher to higher, how to select our experimental animal also is important. It is like that when you are going in any zoo, we know that which animal, how they behave, right? People may go for the monkeys even, but to handle them, it's really difficult. I have worked on the mice, somewhat lesser, rabbit more. To hold such mice, because it is in cage, right? Out of cage, when you are taking out, they having their nails and everything. It's like that someone is trying to control you, give the injections, so you will start to fight like anything, right? It's required to be. So when you are injecting in, and another is same animal, we have to maintain at least for the 6 months to 1 year, depend upon the kind of animal is there. So we need to take their bloods. I think so. Yes, you are telling about some clinical microbiology. You was asking that after doing your bachelor's, if you're looking for particularly into some directions, it is like agriculture sector, medical sector, and other are goes on. So there the one is paper is coming, animal physiology and understanding or the handling of the such animals. So on a rabbit, if you are, I'm asking to take your blood from you, from where it has been taken, generally from the our veins. Hence, it's been asked. depend upon the age, right? Hand is taken. If the child is there, from here itself. If the infant is there, if you are knowing, that is from the legs. Towards the handle, just a small hand, it's really difficult. So over the time, such practices is required. So when you are looking to handle our animals, okay, in a rabbit, it is generally been practiced. You are trying to, because they are having the fur on their whole skin and body part. So it is required to remove. Otherwise, been taken from their ears, because they are the smooth muscles. From there, you can start to inject even. But when you are injecting your chemicals or any products, what you are looking as the experimental part, either maybe antibiotics sometimes, or any pathogens you are looking to inoculate to your animals, there may be a swelling on it, or some errors may start. That physiological disturbance will be there. It is like that when you are going to a very good hospitals. Sometimes we are calling that we got the reaction on the whole skin, right? It happens, may happen. Okay. So doctor is not looking to make you a harm, right? He is to elevate your problem, looking to reduce your problem. But sometimes by chance, that is the genetic, you can say the influence. That is the one core areas of pharmacology is coming, pharmacogenomics. That what drugs is being given to us, our genetic response will be a different from one to another individual. That means one person is able to eat or digest any number of the paracetamol is given, and some is the just 250 mg. Generally 500 mg is there. 250 mg. Half of the paracetamol is also given. The person will in a problem. High acidity, headache will start, and then the diuretic conditions. It may happen. So they are not allowed, or they are not looking to prescribe such another kind of a tablets. So all of the points are there required to do. So over that time, when you're looking to work on the animal related any aspects or the viruses or media, then we need to go for the selection of the nutritive material that is from the extracted from the animals. So when you are looking for the gelatin or the gelatinous product, okay, that is from the bone and cartilage are being prepared. Okay, next it is, there is a agar. Mainly in our microbiological part, specifically about 80% you can consider, 70 to 80% is solidifying agent will be the agar. It came from the one known algae that is like a Gelidium corneum. There are the several, 2 to 4 are available, different kind of a you can say the brown algae are there. From this brown alga, when it is been grown, it is for them, it is one kind of a polysaccharide. Okay, kind of a polysaccharide, it is been synthesized. And this polysaccharide, we are looking to extract it out. So what is the basic property? When you boil it, it is going to be generally get dissolved. After the dissolutions, if it happens, when you are going to keep for in a static condition about half an hour to an hour, it is gets solidified. It is like a gelling material. Generally, this agar also been used in so many cosmetic and beauty parlors also. When you are looking for in beauty parlors, such things are already been added. When the within a temporary time period, you want to make some jelly material or solidifying. Here the best property is what? If you are going to add the agar in a higher amount, that is a certain limitations and part is get, like generally it is 1.5% to 2.5% is a amount, optimum amount is given. It is like that adding the salt or something. It's going to solidify. 2.5 gram of our agar powder can solidify about 100 ml of our solution. If you are going to add somewhat lesser, it is like you thought that let me save it. So you may add the only 0.8 gram, solidifications will be there, but the material will prepare like a slurry. It is like a cementing material, right? So if you are going to add more in agar, let's have it in higher amount. If you are going to add the same amount, let's take example 4% or a 5%. What was required 2.5, you have added double. If you are going to add the double the amount, solidification is very high, thicken up. Now you are looking that our microbes should able to grow. It is like that. If some of the parts, yes, when the sabji is prepared, disease, right? So some diseases are required that oil content is given in some of the amount, and the water content is also there. If it is in dried form, you won't like it. Some of the amount is required. It is like from the cabbage, Manchurian is prepared, the process will be different. And some other, you are making the from the cabbage, some dishes are, then it is will be different. But the same thing, just one change will be there. Microbes are here our main part. For us, that was a different is of food. So when you are growing on a 5% agar, the microbes is required to start the nourishment there, and they is able to survive and settle. So for their settlement, it will be very much difficult because they require to, and yeah, we are generally talking for a bacteria more. But as a microbiologist, you know it required to remember that not only a microbial part is coming as a bacteria. Other is a fungi is also there. Algae. So throughout the world, more attention is given to the bacteria. Other are there, just people are forgetting. Okay. So bacteria is easily available in part. Second is the fungi. So even when you are looking for like a PDF, potato dextrose agar is there, one of the kind of is there. So it is being used for the fungi. Fungi very much like potato, like a kids, right? So in a childhood, it is very much loud, right? Even now, in our especially in our Gujarati dishes, half of them in the houses, potato will be there. Otherwise, this will be not completed. So like that only, potato dextrose agar is there, right? It is very much loved by fungi. So there also gelating material is required, or the solidifying agent is required. The agar is there. So fungi will start to develop their mycelium. They will start to develop their root-like structure. That's called the mycelium. This mycelium, if you increase or decrease, right? It is like the plowing is done on the soil. Before the sowing the seed in any field, every farmers go for the plowing. એટલે કે ખેડે છે. બરાબર છે. ખેડવાની જરૂર શું છે? What is the need of the plowing? That when I'm going to sow my seed in any farm, the plants should able to develop their roots and easily able to move. If it is not plowed well, then the root development will be not there. So it seems like that solidifying agent, if it is in twice in amount, fungi is not able to grow very easily. So percentage is very much essential. Why I'm telling twice and thrice, such thing is not been written here in any books. It is a practical aspects. It is a like a knowledge is information converted knowledge based on the experiences. Sometimes when you are coming in the lab, 5 groups are working. It may happen that you have added everything in the certain amount, even though you will say that, sir, our media is not getting solidified. Now, right now, I'm not going to give the what are the problems and issues could be there or may arise. I will ask you people to identify the problem over there. If you are not able to, I will help you out. But it's going to happen that you are going to say that, sir, we did what you told in the same manner. It is like the making a chapati or the making a dish at the home. Every individual having their own way to process and work out, and the taste will change, right? But and it is served to the everyone is eating. But here it is for the bacteria, okay? And the people have started individually or in a group. The bacteria will not like. They will say that we are living in luxurious life. We need certain things to be get catered. If you just miss something, it may be some other issues also. That part I'm not right now going to tell. In the practical sessions, many problems will arise. You will say that I will ask one by one. Okay, then you will say that we did this. That means what are the do and don'ts are there that you are going to learn from there itself. Right. Yeah. One by one. Okay. Sorry. Okay. One question. Yes. Can you be louder? If the fan is going on, so I'm not able to. Yeah. For the agar also, just I haven't talked, just I have, in brief I had talked. Okay, whole process may be not written here. In practical part, I'm going to discuss, but let me brief it. One question was, what is the need of solidifying agent? Regarding the, your is the technical part process you are asking, right? So need of the solidifying agent is first, let me clear. Then some of the part, when you are looking in the working out, it in liquid broth possible. Okay, bacteria can be grown in the liquid conditions as well as in solid conditions. So fungi is not easily able to grow in a liquid conditions. They require static condition and the support. It is like that we cannot float in the air. We need some base, right? Fungal spore can easily, bacterial spore can easily. Okay, from the moisture, bacteria can grow very easily, not fungi. So fungal, or that means what kind of the bacteria or your organism is there, accordingly we need to follow. Okay, now the second question is about him. That's regarding the what is the solidifying and the mechanism. Generally, it's a, we are boiling at 100-degree temperature. Agar is been added into it. It is near about a 55 to 60-degree temperature is start, it will start to solidify. So while handling and holding our special agar is dissolved, then we are going to autoclave it. After the autoclaving, when it is coming from the autoclave, its temperature regarding about 121 degrees centigrade. So you are having about 20 minutes, 10 to 15-minute time period, because when you are taking out of the autoclave and you have kept near on the certain on a desk, right, it will start to get cooled down. So you need to complete your task maybe in a 15 to 20 minutes. Otherwise, it is going to be solidified within your flask. Okay. You alternately you can go sometimes again for the boiling. You have that solidifying agent is there. You may go for the boil, but twice or the thrice if you are going to boil again and again, then the capacity of one is like agar, and the second most important part is our nutritive ingredients. It is been added, it start to get deteriorate. It is like sabji you have prepared at a morning, or it is like some people are in a here or so many foreigners are there, less time is there. For a week, they have prepared one dish, right? Sabji is prepared, kept in the freezer. Every day they are taking out for as much as the amount is required. They have the micro oven, just heat it, they are taking. But the same things, you have heated micro oven, you have kept for a while, some phone calls has came, after hour you are coming, the food was just cool down. You have again microwave. Size if you are doing, then you are going to eat it, you will say that today is not good. It's like a tea is there. When you are going to canteen, you are telling that, oh, that person has made twice and thrice. બેથી ત્રણ વખત ગરમ કરીને આપી દીધું કાઢેલ. The taste will change. The ingredients, what are there, it start to get charred. The term is charred. That means they start to get deteriorate. That means either there is a breakdown of the original molecules may start. Continuous boiling and heating is going to reduce down the capacity, or the ingredients what are there, they are going to get deteriorated. I hope it's clear. Your question is yeah, fermentable compounds. You got the question answered? Okay, solidifyingly. Okay, fermentable compounds. Yeah, another part. That some bacteria or the fungi, we have talked about the lithotroph, autotroph, like heterotrophs are there. They required half of the things been ready. They are not going to grow on the nutrient broths. They required facilitated. Some things should be get facilitated from the beginning itself. Then only they are going to grow. So some are like monosaccharide, glucose, fructose, disaccharide, polysaccharide. So many things could be possible. Okay, so such thing is required for them. So fermentable, it's like a fermentation term is going to come. You need to keep in your mind, that is, we are going to look about the aerobic bacteria to anaerobic bacteria then after. Okay, fermentations not always anaerobic. Don't put like that. Fermentations process, we are, I think you may have the one paper, bioprocess technology or fermentation technology. So there you are going to have the larger vessels for the fermenter. You are going to have the agitators also. Aeration, agitation is done. Okay. So they regarding the aerobic also and the aerobic and aerobic also. But it is regarding the fermenter. Fermentation, when we are looking, it is related to anaerobic conditions, alcohol going to be get at the last product. And aerobic condition is there. So many bacteria are there. So what is the basic thing is there? They are not started with all basic compounds. They require further nourishment directly chewable or eatable items. It is like a person when it is hospitalized. Through the veins, nourishment is given. Otherwise, the glucose powder. They are being offered with the chocolates or the choco ice cream or others because they are not able to eat or the chew or the digest like a chapatis. If they are being given, let's say the pizza, he is in hospital, so he liked it, he or she is not able to take it because for that energy will be required more. If you are going to give the butter and everything will be higher, he going to get the diarrhea either or vomiting. Okay, so in that conditions, something which is digestible like that, some selective bacteria are there. For them, we need to go in that format. Yeah, now the classifications will come regarding the medias. Okay, consistency, that is now our main part will start in a way that selective media, supportive media, or what are others. So first time it is classified as solid, semi-solid, or the liquid medium. Three are there. I think, right, let me close it. The solid, that is related to nutrient agar. Okay, nutrient agar is there. Then the solidifying agent we have added already. One another is a semi-solid, and third one is a liquid. So I just told about two main. One was regarding the solid, another for the liquid. What could be the possibility of utilization of a semi-solid? Kept on water, so many are possible. Semi-solid, just if you're able to think about it. I have told about what? Solid, then the liquid. Now middle one is remained semi-solid. Eh? No, consistency, it is regarding the state. Consistency, that is going to remain in a one state. Constant in a solid state or in a liquid state or in semi liquid state or a semi-solid. Can you be louder, sir? Okay, preparation. Okay, that's right. In preparation, so semi-solid means you have added agar powder in lesser amount. Solidifying agent is required. I'm asking for the applications. That means about 2%, 2.5% is required. Then it will be get solidified. Okay, but you have added just suppose 0.8% or the 1%. Then it is became semi-solid, right? Where and when is required to use? Okay, right. Partially, but not fully. Here, in liquid medium, nourishment will be there. In solid medium, nourishment will be there. Even in semi-solid, nourishment will be there. So all your answer is satisfied, but the real answer, application point of view, what been asked is not yet been. Here you are telling that agar will be lesser, right? But we are not looking for the saving here. We are, yeah, in Marwadi we are, so saving mode will making, or wherever it is there, right? Economic point of view. That's not my point to be asked. My point is regarding the applicability. ક્યારેક આપણે ઘરે ડીશ બનાવીએ તો અમુક વસ્તુઓનું આપણે સ્ટોરેજ કરવા માટે યુઝ કરતા હોય ઘણી વખત ઘણા એના બીજા પ્રોસેસ થી કરતા હોય પેકિંગ માટે મૂવમેન્ટ માટે just think like that. It is like cooking. If you are able to integrate those who are, that's from my guide and so many, you are going to heard such things so many a time. A good microbiologist is a good cook always. And if it is not there, you may start to learn certain cookings at your home. not for a daily purpose, but at least then you are able to understand all the phenomena. Not right now, but here in later also. If it is you are working in certain laboratories, just it has been told that we want to do such work. Nobody knows because in the world no one has done. It's like a coronavirus, something has happened, or the some of fungi. It was it has been mucormycosis. Nobody has worked in a world a lot on it. It is the task is that you are working in the good laboratories, you are having the awesome amount of your package, and the task is given. Now you may start to do the work, but how to do a handling or part? It is like a something is there, the subject is given to someone, right, to cook. First day, it is like a 10 kg is given. First day you may start. You may found that there certain vegetables is very hard having their outer skull. So you will start to you find the some utensil or the some cutter or the some knives to cut it down first. The first day you come to know, okay, it is having the some problems that you have solved. Okay. So same day when you are continuing a daily 10 kg material is there, how to cook or the prepare it, because maybe you don't know whether it is a having the bitter taste, sweet taste, some vegetable is with you on. In mixing where how you are going to. So same thing is going to happen into all the categories here. Which kind of this our requirements? It is like that some guest is coming. Yes, jelly like. That's true. That's what also I'm telling. You can also, I'm giving, yeah, search it out or try to discuss among yourself for a 5 minutes. Applications of which I will among yourself. Application of solid media, liquid media, and semi-solid. You can have the discussion among yourself also. You can search it out if you require to use your digital any gadgets. I never mind. Just search it out. Application of which solid media, liquid media, and semi-liquid. You can have the discussion among yourself even. Okay, search it out. After it is like 8.2 is there. 8.4 I will or 8.5 I will ask. Those who are in online mode also, search out the application of say solid media, liquid media, and semi-liquid media. It's a kind of a brainstorming for you. So you have the discussion among yourself, or you can thought by yourself. You can use the certain gadgets even, not a problem. So I hope you got the enough time. At least those who have found something, we can have the discussion. You can search it out. Yes. What is motile, non-motile? For what? You can yes near to it, but yes, just focus more. Be louder somewhat. Motility નો ડિફરન્સ મતલબ સોલિડ અને લિક્વિડ. હા એ તો છે, લિક્વિડ હશે એ તો સરસ મજાનો ગોળ ગોળ ફરે, સોલિડ ઇઝ સ્ટેટિક. ઓકે, સેમી સોલિડમાં now you got the difference. So any applications no થાય. I'm asking for the applicable applications of each. ક્યારે મારે સોલિડ વાપરવું જોઈએ, લિક્વિડ વાપરવું જોઈએ, difference is now understood. Well, very nice. Okay, that is also, he is also right. Motility. Second point is there, that is a colonies we require to understand the characteristics are there or in it. Liquid, bacterial population. Now the third segment also required to understood when you are going to use the semi-solid. Search it out for that also. What are the applications of it? There are enormous number of both solid and liquid applications are there. Still you have just told one or two only. The also one important segment we haven't yet discussed about semi-solid. What is the applications of it? એટલે કે થોડું ઓછું છે. જેલી મટીરીયલ છે, સોલિડિફાઈડ એટલે એકદમ કડક હશે. આ જેલી છે, જેલી મટીરીયલ ક્યારે વાપરાય એટલો જ ક્વેશ્ચન્સ મારો છે. Maybe what kind of conservation? સેમી સોલિડ ની એપ્લિકેશન્સ ક્યારે વાપરવું છે ઇન માઇક્રોબાયોલોજી? Motility. Okay. How you are going to measure the motility? But so be louder somewhat. Then in solid or oh, no, because bacteria will not start to move in that way. They will okay. Not that, that's not directly. Maybe you are near, but in explanation point of view, it is not true. Maybe it is related to it, but explanation is not in that sense. Halfway is somewhat maybe near to this, but it is not completely confirmed because you have seen superficially, not understood properly.
System, how motile we can function on semi-solid medium, and then even for isolating the bacteria for like if you want to purify the bacteria for pure culture, you can go for semi-solid.
No. Okay, but for the isolation for pure culture techniques, we may go for the either direct liquid to solid. Solid will be higher than the liquid. We are going for the serial dilutions. That is the most important. Pure culture, I already told some of the times. Then the liquid culture, if you are looking, then you may go for the serial dilution of the soil sample up to 100 to 10,000 to 10 lakhs. Then you are going to get the colonies well settled on the solid. Not about the semi-solid. On semi-solid, we are not going to do that part. There you have to dwell your mind, you have to thought about it. Till you're going to find, that means you are going to understand the both the places, when to use the semi, when to use the liquid. When it will be get clear, then only you are able to understand the semi-liquid, semi-solid. આ બે સમજાશે ને તો ત્રીજાની ખબર પડશે. Yeah. Challenge my. Huh? Yeah, so that is true. You understood well. But when to use semi-solid, colony is not going to develop properly. And the liquid medium is also like a swimming. bacteria start to swim. Somewhat there, swimming is also there. It is like that you are having the something different kind of a going in a kindergarten, some special ride is there, right? The purpose of the special ride is the amusements, but daily you are not going in such buses or car, right? It is a ride, so you may go for the walk or something is there, so it is the something different. So what could be the applications, when to use, where to use, how to use, what could be the purpose? Yes, yes. ક્યાં છે? Okay. Okay. શું isolate કરવું? Somewhat near to right. There are the on just it is not main applications. Let me okay, there is not a somewhat here and there motility people are telling. It is not only a one. It is a alternative applications. You will find. I will ask tomorrow. Note it down in your diaries. Okay. Yes, this is required to. Otherwise, you will going to get the answer tomorrow. No problem. That is up to you. Note it down in your diaries also. Applications of semi-solid medium. I thought you understood what is semi-solid, how to prepare it. The agar considered solidifying agent is somewhat lesser. That's the main part. Nothing. But what could the applications? Yeah. So that part I will discuss later only. Let's move ahead then. No problem. We may discuss in at 10:40. At 10:40, we are going to have our lab today. Okay. So I will ask you first to be here. It is not there. Okay. It will be tomorrow. Chemistry will be tomorrow. So I will take you people to the lab first. Before, let me complete. Then you are having to have the answers. I can tell. Okay. So I will ask you people to do some calculations over here. Then we will directly move for the media preparations. Today we are going to prepare the first, that is a liquid medium. I will ask you the number of students. How many is there? Someone can calculate, count. Huh? 19. So at least we have the 4 groups. One group may have the 4 students. Another group have the 5 students. So among yourself, you may distribute yourself during the time of after the break. I will ask you for the first you people some calculations. Okay, some virtual part is there that we are going to observe and learn. Then we'll go in the lab. We are going to understand about the where the utensils are there, what are the basic steps are there that you are going to perform today. Okay, into it, right? So if you are going to get the time between it, if you are able to find the answers for the semi-solid, we may discuss it at 10 or 10:40 in between a break that time. Otherwise, we are going to discuss during that in a lab condition, because it will be after the preparations, everything tubes land, everything wrapped, it will be autoclave. During the autoclave time, about half an hour and hour will be there. We may adjust in a way that it can be used in a form of a break. You can have the lunch break. You came back, we may go for further steps. So within that time, we can discuss about the topic semi-solid or applications are there. Okay. So we can save our time. We can use our time. લેબ અવર્સ તો આપડા 10:40 થી હશે ને 1:30 વાગ્યા સુધી. એમાં અમુક સમય તો તમારો પ્રિપરેશન્સ માં જોઈ જશે. એટલે શરૂઆતમાં અડધો પોણો કલાક કહું છું ને તમારે પહેલા તો કેલ્ક્યુલેશન્સ કરતા શીખશું. ત્યાં જઈને શું કરવાનું છે તો ખબર છે? એ નથી ખબર. બરાબર છે. I will just going to say, okay, that prepare 15 tubes, each group for 20 ml N broth. If you are able to do, you may do it. We directly move at 10:40 or 10 tubes. 10 tubes. Each group are going to prepare only maximum 10 tube on. 10 tube nutrient broths we are going to look to prepare, and each should contain about ml, 8 to 10 ml, not more than that. So that means you are going to prepare 100 ml nutrient broth. How you are going to prepare that I don't know, because you told that you know. No. She is telling that we can go, we can do, right? No, because you have started to give the reply first, so I thought that it is you are knowing. That is why I'm telling that what you are going to do in a lab, that I'm going to say first, then you need to do certain calculations over it, then you can able to perform. Okay. Okay. So another, it is like a, okay, some has put in the chat box even, let's see. Isolation and colony characterization of bacteria, semi-solid transport liquid. The liquid for the growth of microbes. Okay, someone is telling that is related to transport. Who is there? Yeah. Celestine Salema. Okay. Yes. Can you, Salima, can unmute yourself and say about how to transport it is being used? What is the basic things related to transport? I think Salima is girl or boy? No? Girls or boys? Okay. That fellow. I think he is writing down the transport, but maybe not. Sir. Okay, I will continue at 10:40. Okay, ending here at 10:40. We are going to meet here again. We'll do some calculations basically, and additionally, I will give some few examples of the questions related to the solidifying medium also. That is like you are required to prepare a 10%, 2%. So some problems related to percentage. Okay, so maybe you are having some diaries with you, so you can note it down over there. In exams also, in a Viva point of view, or many times, same problems can be asked. Okay, that means in a theoretical part also, even in your objective somewhere, it can be asked how to prepare the 2% solutions, 5% solutions, or it been asked in a way that you require to prepare 750 ml 2% solution of a glucose, how you will prepare, how much amount is required. Okay, it is for the 100 ml. If I'm asking the 2%, that means 2 gram. If I'm asking for the 750 ml, then what and how you are going to calculate, that is up to you. So such basic part I will ask first, then we'll go directly in lab.